Welcome to the studio and welcome to this company presentation. With me I have NextCell Pharma's CEO Mathias Svahn. Welcome.
Thank you very much.
And you have a whole array of staff members as well. We have Per-Ola joining us in a second, and also Lindsey Davis with us on link. But you're gonna start with the presentation, and then we're gonna hand over to the other staff members as well, and we're gonna end this all with a Q&A session. That's the entire reason I'm here. So for viewers joining in, please write your questions in the live chat as well. I'll be sure to bring them up. Over to you, Mathias.
Thank you very much. I'm very excited to be here and proud to be able to present the data from our clinical trials together with our principal investigator, Professor Per-Ola Carlsson, and our scientific officer, Lindsey Davis. But to start with, here's our disclaimer. So, NextCell Pharma, we are a Swedish company that has developed a cell therapy treatment to modulate the immune system in autoimmune diseases. Our drug candidate is called ProTrans, and our first indication, the first disease to be treated, is type 1 diabetes. We also have two other projects where we are evaluating ProTrans. One project that is ready to go into clinical trials, and we have yet a third project that is still in the preclinical phase. These projects are all based on our drug candidate ProTrans, which is a mesenchymal stem cell therapy. We also have a stem cell bank, or cord blood bank, called CellaViva, which has grown to become the largest cord blood bank in the Nordics. Our drug product ProTrans is primarily investigated in type 1 diabetes, and this is our clinical trial program. We have conducted a phase 1 trial, a dose escalation trial called ProTrans 1. And we recently got the data from ProTrans 2, which is our placebo double-blinded randomized trial. And we also have a repeat trial where we give a second treatment with ProTrans in the ProTrans repeat trial. And we plan to evaluate our drug product all the way towards patients in the market. It's a great study team that we have put together that has been able to conduct all these trials within the time plans. Professor Per-Ola Carlsson is the principal investigator. He's from Uppsala University. And the co-investigator is Daniel Espes, also from Uppsala. We have gotten huge help from Dr. Sofia Cissé from Karolinska Trial Alliance. And we have a data safety and monitoring board with professors from both Gothenburg and from Linköping, Ulf Smith, Anders Fast, and Åke Lenmark. And in the NextCell team, we have our Professor Edward Smith, who's the Chief Medical Officer of the company, Dr. Lindsey Davis, Chief Scientific Officer. We have Dr. Hegg, who's the Quality Manager, Dr. Forsell, who's the Laboratory Manager, and then we have research engineers as Bahar Kalai and Carla Westin. So it's a great team, and I'm very proud to be able to welcome Per-Ola Carlsson, who's the principal investigator, to take us through the data.
Okay, thank you, Mathias. I will talk about the primary endpoints of our ProTrans study that we have recently been conducting. To start with, ProTrans 1 is an open-label dose escalation trial of Wharton's jelly-derived allogeneic mesenchymal stromal cells, or stem cells, as Mathias told you. And we aim to preserve endogenous insulin production in type 1 diabetes, and this trial started out with adult patients for safety reasons. Inclusion criteria was that they should be diagnosed with type 1 diabetes during the last 2 years. They should have still some remaining C-peptide, and we define that as a C-peptide above 0.12 nanomole per liter. They were supposed to be between 18 to 40 years and have a body weight between 50 and 100 kilogram. They should be otherwise, besides the diabetes, healthy patients. These are the baseline characteristics that we obtained. We started with a dose escalation part then and included 3 patients to treatment with a low dose of ProTrans, that is mesenchymal stem cell preparation, and that was 25 million cells. Then we treated 3 patients with a medium dose of 100 million cells and 3 patients with a high dose of cells, and that is 200 million cells. And as you can see, they had an average age of about 24-25 and 27 in the high dose, but that was not significantly different. They had a diagnosis for an average of 1 year. They were all males in this first part, and the reason for this was that we were a little bit concerned about if we would have HLA immunity, and that was something we also studied and did not see in the end in this part. The body weight was about 75-80 kilogram, normal BMI, and then we looked at how much insulin they had to start with, and that was conducted as an area under the curve for a mixed meal tolerance test. And when we calculated this area under the curve, we could see that was a slight difference, meaning that the patients that started out with a low treatment, and that was the first 3 patients, they had slightly higher remaining C-peptide than the rest of the groups. This part, the dose escalation, had a primary safety endpoint, of course, because we wanted to see and investigate the safety of these 3 different doses. And the safety parameters included adverse events, of course, and some specific studies of hypoglycemia, allergic reactions, ophthalmology to look at the retina, and also different laboratory assessments. And in the end, we could show safety in this part. We did not see any particular changes compared to normal. And then we also had the opportunity, of course, to look at the efficacy of this treatment, and we investigated that as a change is in this remaining insulin production from at start, just prior to treatment, and at a follow-up 1 year later. And then we could see that those that received the low-dose treatment, they lost in units about 30 to 40 nanomole per liter over 120 minutes, and in percentage they lost about 30% of their C-peptide insulin production during the first year, while the patients, individuals treated with a medium and high dose of cells, they did not obviously lose any insulin production at all during this 12-month period, and the mean was about 7 to 8% loss during the 12-month period. And that was a significant difference actually between these groups with the advantage of a treatment with a medium and a high dose of cells. And since we had safety in this first part, we went into the second part, which was a randomized and blinded part where we compared 2 different batches of mesenchymal stromal cells to a placebo. And that meant that we had 5 persons in each group, and therefore 10 patients were treated with ProTrans, while 5 patients were placebo. This was a very small phase 2 study, of course, but we still thought that we had a significant effect in this already in this dose escalation trial, and therefore we aimed for this small study. And in this study we showed that we used the same criteria. They should be diagnosed within 2 years, have a remaining C-peptide, the same age, the same weight, and be healthy patients. The only difference mainly was that we now also included females in this study. And both the investigators, nurses and doctors, and the patients were blinded to the treatment, and therefore we could evaluate to placebo, and this was studied throughout the course of 1 year, both with a safety endpoint, looking once again at adverse events, the same as in the dose escalation part, but also with a primary efficacy endpoint, meaning that we, as in the dose escalation part, looked at the delta change of C-peptide area under the curve for a mixed meal tolerance test at 1 year when compared to the same investigation performed before treatment. And in this case, the baseline characteristics were they were about 31 years of age. They had a diagnosis 1 year ahead of inclusion to trial. Both males and females, about 50%, were included. They have a body weight of about 70 and a BMI was similar, about 23-24. Also in this case, there was a difference in the baseline by random of insulin production at baseline. But in this case, it was the treatment groups that had more remaining C-peptide than the placebo at start. Then we looked at the difference in percentage between the C-peptide production in response to a mixed meal tolerance test at 1 year compared to before treatment, and showed that while the placebo group lost a mean of 37%, the ProTrans treated group only lost about 10% as a mean during this year, and that was a significant difference when tested by a two-sample two-tailed t-test. It was a p-value of about 0.01 actually. Okay, I'll invite Mathias back to look at the meta-analysis.
Thank you very much, Per-Ola. So I just wanted to highlight a couple of features from our trials that we see with ProTrans being active in both these trials. As Per-Ola was talking about, we do see differences at baseline C-peptide, which is the patient's own insulin production before treatment. And in this picture, we have both the trials, ProTrans 1 and ProTrans 2. And as you see in the bar to the far left, that the low patients, before treatment, they had on average around 130 units, which decreased over a year to around 90 units. So they were falling relatively much from a relatively high level, whereas the medium and high dose had a significant, a substantially lower limit or lower level of insulin production before treatment, but they also lost very little of their insulin production 1 year after treatment. We had the reversed in the ProTrans 2 trial, where the placebo patients started from a low level and they lost a lot of their insulin production, whereas the patients in the ProTrans treated part had a much higher level. So we can see that irrespective of baseline C-peptide, we do see impact with ProTrans and efficiency in these patients. The tendency here is that the low-dose patients and the placebo patients are similar in their insulin reduction, whereas the medium and high dose in ProTrans 1 are not significantly different. And the high dose in ProTrans 1 and high dose in ProTrans 2 receive the same kind of therapy. So this is exploratory analysis, but here we compare optimally treated patients, the ones that got medium and high dose, with the patients that got placebo or low dose. And when we group these patient groups, we see very nice data where we can protect the insulin production in the patients treated with optimal doses of ProTrans. And this is seen both in absolute values and percentage. So a lot of figures, but there's also patients behind these figures. So looking at the patient outcome, we have 4 out of 15 patients that had an insulin increase during 1 year. 4 out of 15 in the optimally treated patient group, whereas none of the 8 patients in placebo or low dose had any increase. And also when we look at the level where we have almost no difference, maximum 5% decrease, we have 7 out of the 15 patients ending up having almost none decrease, and none of the patients in the placebo were able to have that levels remaining. And when looking at a 20% decrease, we had 12 out of 15 patients and only 1 in the placebo group. So this is of real value, and we want to investigate this further, of course. We have been talking about a ProTrans 3 trial, and are now discussing this with CROs, and we will consult the regulatory authorities to design this trial so we can achieve market approval if it is positive. Our anticipation is to have this with the same kind of inclusion control, and the scale of the trial would be in 100 patients. So we do have a full program. We have conducted dose escalation and randomized. We have an ongoing repeat trial that we hope to be able to present later this year. We are planning for placebo randomized controlled trials, and we are also looking at possibilities to support investigator-initiated trials to expand the usage of this treatment. But our priority is to bring this ProTrans to patients, and we believe that the best way of doing that is to have a ProTrans 3 trial with the same kind of inclusion criteria to build on that data. We see that there's possibilities to use this treatment for other autoimmune diseases and other inflammatory states. And we also want to expand the label of ProTrans to also treat younger patients, perhaps be able to treat patients repeatedly, and also treat patients that have had their diagnosis longer than the 2 years. So by that, I would like to present Dr. Lindsey Davis, who is our Chief Scientific Officer, to tell us more about how ProTrans works.
Thank you, Mathias. So a part of our understanding the mechanism of action of our MSC product is essential to ensuring patient safety, but also reproducibility and clinical efficacy of our product. And in order to refine our clinical protocols further, we need to understand the best dosages for usage, their delivery methods, and the role of the patient milieu on a per clinical indication basis. Mesenchymal stromal cells have been exploited for their inherent anti-inflammatory properties in the treatment of numerous autoimmune and inflammatory disorders, and these include type 1 diabetes, multiple sclerosis, graft versus host disease, and most recently a couple of trials for COVID-19. Within the context of type 1 diabetes, this is an autoimmune disorder affecting both the innate and adaptive immunes of the arms of the immune system, resulting in destruction of the beta cells in the pancreas. So knowledge with respect to how ProTrans itself interacts with these particular immune cells upon administration will help us to further develop our product and further refine its usage. Can I have the next slide, please? ProTrans itself is an allogeneic MSC product, as we've discussed, and it's derived from the pooling of ex vivo expanded umbilical cord mesenchymal stromal cells that have been derived from the Wharton's jelly component of the tissue. The cells are initially defined according to guidelines set out by the International Society of Cell and Gene Therapy, but their unique selling point comes from their combination of a GMP-compliant and xeno-free production protocol, which in combination with our patent-pending selection algorithm allows us to evaluate and select the highest quality MSC donors for pooling and generation of our ProTrans product. The next slide, please. This algorithm allows us to reliably reduce batch-to-batch variability between our products themselves, and also to ensure a high quality and functionality to ProTrans. We have developed in-house a battery of functional assays as part of our MSC donor selection process, and these have been designed to maximize the potency of the MSC product. As you can see from the graphs here, with testing of 3 different functional parameters, we see large donor-to-donor variability between the MSC donors themselves. But with application of our selection algorithm and pooling to generate our ProTrans product, we see minimal batch-to-batch variability, ensuring that we have a robust and upscalable product. Next slide, please. So to conclude, our ProTrans clinical production protocol, in combination with our selection algorithm, ensures that we have a potent and functional product that can be robustly manufactured in large batches and with low batch-to-batch variability. The allogeneic nature of our product means that it can be produced in advance and offers this off-the-shelf availability to patients and clinicians. With a proven safety portfolio and easy administration at the bedside, ProTrans offers the potential to be developed for usage in the treatment of a wide variety of clinical indications. Using umbilical cord tissue as our cell source of MSCs, we have a potent cell source that rapidly expands in vitro and allows us easy and non-invasive access to the tissue as it's discarded after birth, which offers distinct advantages over other MSC sources on the market, such as bone marrow. Thank you. Back to Mathias.
Actually, I'm gonna grab the word right there, because it's time for the Q&A. I've invited back Mathias and Per-Ola back to the studio. And Lindsey, I'm gonna ask you to stick around, because I think I have a few questions that only you can answer to. But I want to start with Per-Ola, actually. If you could just simplify this, because we've gotten a lot of information now in just these last 25 minutes or so. Put it in as simple and as practical terms as you can. What do these results mean for a type 1 diabetes patient?
Actually, if we are able to sustain the remaining insulin production at debut, they have about 20, 30, 40% still left of their own insulin production. And if we are able to stop the further destruction, they will have a much milder diabetes, so that in the end, if they have much milder diabetes, they will have much better blood glucose control, and in the end, they will not have complications to their diabetes and have a much easier handling of their insulin administrations. In the end, we could also say that there are screening programs to identify individuals at high risk of developing diabetes. So it could be envisaged that in the end we could give this to individuals that have sign of autoimmune disease but still have not developed the diagnosis of type 1 diabetes.
And the next question I think I'm going to ask to you, Mathias, but you can grab the word as well, Per-Ola, if you want to. How sure are you that these effects that you discovered are long-lasting?
We have proven that they last for 12 months, and we will, of course, like to prove that they last longer. This is fantastic data that we can stop disease progression for a year. And we have, as Per-Ola said before, this is a limited trial, and we need to investigate this further.
And if If we look at the scale of the study, it's only 15 patients. To me, that's a small amount. What about the reliability and the significance of the results? What can you talk to me about?
The weakness is, of course, that there are few patients. On the other hand, it is a substantial strength that we still get significant results with these few number of patients in 2 parts of a trial, meaning that the effect is very reproducible, seemingly. So I have good hope that we will be able to repeat this in further studies.
And what about the possibility, I'm not sure who I'm asking the question to once again, the possibility to treat younger patients or patients that have had diabetes for a longer time?
I could answer that because younger patients, I think that is something that we aim for in the end to also be able to treat. And mesenchymal stromal cell stem cells have been used for treatment of other diseases in juvenile patients, and have shown no obvious side effects there. So I think it's fully feasible to go into that age group too. So that is no problem. To treat individuals that have diabetes for many years, that might be a problem if they already have lost all of their insulin production, because this is we haven't tested it, at least. It does not show that we can regain insulin production in those.
And Mathias, one of the most asked questions, I've reached out to social media and I've seen it in the live chat today as well, is specifically the phase 3 study. How would it look?
So we are discussing that with the CROs, that is a contract research organization that would help us with a multi-national trial, because we would then have to run a trial in more than countries than Sweden. We will talk to the regulatory authorities to discuss how to design this trial so they are able to prove it. So this is something that is an ongoing discussion. We believe that our best aim is to bring this treatment to the patients that we already shown both safety and efficacy in, which is then adults that were diagnosed the last 2 years. So I believe that we will keep the inclusion criteria, and then we have to discuss how to maximize the data for the phase 3 clinical trial.
And what about the scale? Multinationality sounds a lot bigger than 15 patients at least. Can you talk to me about how many?
In cell therapy, the patient numbers are not that high. It's not like bringing a vaccine to the market and you try it on 30,000 patients in a phase 3. In the scale of 100 patients is a fairly large trial in cell therapy. So it's not compared to small molecules and vaccines. Of course, it's going to be a larger trial, and it's also a strength for us not to have only Swedish patients and not only have one site. So we want to increase that. But when looking at coming back to the small trial, yes, it was rather few patients. It was not very few. It was rather typical for a phase 2 in cell therapy. And when we design a trial, we look at, okay, what do we expect? What kind of outcome do we expect? And then we put the power so we have the number of patients to match that difference. And so one could say we were rather self-confident when doing only 15 patients, but it paid off. We can do it with fewer patients, which is good, and we can bring this product faster to the patients as well.
So my thinking asking about the number of patients is related to costs, of course, because financing is a big part of doing research as well. What do you think about the cost involving 100 patients? Do you have the financing prepared for it, or do you look for a partner, or what's the plan?
We are investigating different options, of course, and my aim is to bring it as fast as possible to the patients, but also, of course, to assure that our shareholders are benefiting from this development. So we are looking at different options how to proceed.
And do we still have Lindsey on the link? We do. Perfect. Okay, Lindsey, sorry. There was a lot of talks about numbers here, about money as well, but I'm going to move it back towards stem cell therapy. It's a rather complicated manufacturing process, and complicated usually means expensive as well. What about the ability to scale production today, Lindsey? Can you talk to me about that?
Well, we've got a very clear GMP compliant production protocol through our contract manufacturer organization, our CMO, that we've been able to refine to the point that we have low batch to batch variability. And that's kind of central to us when we're talking about upscaling, because we need robustness and reproducibility of our product. So we have the ability to now have that production protocol at multiple sites, if that's how we decide to go. But we're also forward thinking for the future for large scale commercialization of the product. And therefore we have recently been awarded a Eurostars grant to investigate switching to bioreactor-based production of the stromal cell product.
Now, Lindsey, the reason I asked you about scalability is that the area of study, based on ProTrans medicinal properties, what other diseases could be suitable for treatment?
So with MSC therapy, they have been used in a lot of academic clinical trials for different indications. Very few of these have shown an efficacious endpoint at the moment. And that is due to a lot of limitation with the passage number of the cells. So that is how much we expand them in culture before we actually utilize them. But they can be effectively used for a number of inflammatory-based disorders, different autoimmune disorders, such as type 1 diabetes and multiple sclerosis, as I discussed. So in emerging acute inflammatory conditions, such as COVID.
Now, Lindsey, there are other treatments that use this same technique, the MSC. What about competition? How does your product compare to the ones already out there?
I think it's important to understand that although different cell sources are being classified as MSCs, the original tissue that the cells are derived from is essential to actually their functionality and their clinical application. And we have a potent cell source coming from the umbilical cord. We have easy access to large amounts of tissues, which means we don't need to expand the cells extensively in culture, which gives us a unique advantage over other cell sources such as bone marrow. But also in terms of functionality of the cells, we see that they're very anti-inflammatory. So therefore, like we've shown in these clinical trials, we're using quite a low dosage of cells in comparison to other cell sources such as bone marrow, which is normally based on a per kilo body weight, whereas we can use a fixed dosage.
Please stick around. I'm going to move over to a few questions from social media and from the chat as well. But I'm going to turn to you guys primarily. About the partnership, what would a dream partner be?
Yes, we are now active in EMA area. So and the company has limited resources. A dream partner could help us having parallel clinical trial programs in different parts of the world. It could also be a partner that is interested in, it doesn't have to be a partner that is interested in diabetes. I mean, we are interested in looking at developing this therapy for other indications. But we would like to see a partner with a larger organization that has a presence in other continents as well.
And you talked earlier about this, the next study being multinational. Is that something that you consider when you choose a partner to partner up with as well? Are there any specific countries as well that you're looking at?
We are primarily focusing on EMA because that's our home visit and also where our expertise is. So we are looking at all the options moving forward, but our primary market is going to be EMA as NextCell. In a partnership, that depends, of course, on the partner also.
Of course. Now you showed us a timeline earlier. And we have one question from social media that I got on Twitter earlier today, and that is, when do you think you will have a product at the market?
Yeah, it's always a tricky question, of course, but we have the ambition to apply for clinical trial during this year. But as I said before, we will not stress this application because it's very important that we get a perfect study design and have the regulatory authority behind us. And then we expect to have inclusion for a year, follow up at least a year. So we are looking at maybe 2024.
All right. Now, I saw that Diamyd Medical, the company, is also in the chat, and I saw a question here. Could the effect from ProTrans, maybe this is a question for you, Per-Ola, could there be better efficiency or efficacy if you combine it with Diamyd's candidate?
It could be, we don't know. There is no obvious synergistic effect between these two products, of course, but since also that product has shown efficacy in phase 2 recently, yeah, it could be worth. But as Mathias said, since we had effect in this trial, I think the phase 3 with one product is what we aim for.
Yeah, it's always hard to start with two unknowns.
Yeah, of course.
So we'll do one
at a time, and
yes.
then maybe combine them after 2024 then. Okay, so Per-Ola, which other treatments are approved or about to be approved as of now?
And that there are actually no treatments to stop the development of type 1 diabetes, and there has been several trials in both phase 2 and phase 3, but there has not been anyone that has been successful or are very close to the market as of now. So I think this is a very strong candidate.
I'm getting questions about a lot of happy emojis as well, but questions about licensing. Is that something on the table as well?
Yeah, of course. I mean, we don't
close any doors at all?
No, of course not. We are open to look at different options. It's all about bringing the treatment to the patients and building value for our shareholders.
I think I've gotten through most of the questions that I've gotten from social media, and I'm happy with Lindsey's answers as well. So I think I'm gonna close the broadcast for now. So I'm gonna thank all of you that joined in and followed us, even if we did it in English today. There were a lot of followers, a lot of activity in the chat. Thank you for that. If you're interested in NextCell and Mathias and Per-Ola's and Lindsey's work, please do continue to follow them. And we'll see you next time we go live.
Thank you.